Hello everyone, welcome to another lecture for drug delivery engineering and principles. I am Rachit and we are talking about some of the tissue engineering strategies and how drug delivery is involved in some of these strategies. So, just a quick recap of what we learned in the last class. So, what we learned is essentially majorly what are the drug delivery aspects in tissue engineering. So, before last class we were talking about tissue engineering and giving some basic concept. Now, we are moving towards learning how drug delivery is used for tissue engineering. And so, in that what we discussed is first of all various strategies to release molecules through matrices, and these molecules can be small molecules, these could be large biomolecules, and then these could be even DNA, which is essentially a part of large biomolecule, or these could be even cells. So, it just basically depends on what is the application, but you can use various strategies to release all kinds of things from your matrices for tissue engineering application. And so, we talked about one simple thing is you use encapsulation. So, you essentially use size, and we have talked about this several times in the hydrogel class, in other polymer matrices class. Is let us say you have a mesh network which has a pore size of 5 nanometer, and if your drug itself is 10 nanometer, then it is going to be staying there, and you can slowly degrade these matrices to release this drug over time. The other is the covalent conjugation. So, mostly we discuss this in terms of growth factors. So, let us say if I have a polymer matrix chain and I want the growth factors to be there for quite a bit of time, what I can do is I can link the growth factors. So, let us say if this is growth factor to my polymer chain through some covalent bond. Of course, one of the disadvantage here is that if the cells want to internalize this or the cells want to see a gradient of this coming through, it will be hard. So, to sort of alleviate that concern, what you can do is you can have these polymer chains which are degradable. So, if these are degradable slowly, so essentially what you will have is, let us say if this is a matrix zoomed out. So, as you go further away from it, you will have less and less growth factor being there, because the growth factor is continually released and going into the sink in the system. So, that will give a gradient for the cells to come in. So, that is one way, but what if the cells really want to come in and also still feel the growth factor there, then what you can also do is you can have something which is a bond that cells can cleave. So, if that is the case, then cells will bind to it, they can then cleave this bond and essentially use this growth factor to then either differentiate or whatever they are planning to do. And then finally, we talked about non-covalent conjugation, quite successful for such kinds of applications here. And so, all it is saying is rather than having the cells to do all the work here, what you can do is you can sort of immobilize your growth factors with some affinity. So, the growth factors can have direct affinity to your polymer chains, and so in that way the growth factors will stick to the polymer chain with some affinity, and when the cell comes in, it may bind with a larger affinity and take this growth factor away. The other option you can do is you can bind the growth factor to a molecule that has affinity to your ECM, and so in that way then the growth factor which is bound to this molecule will attach to ECM, and the cells can still pull it away along with this molecule. That may decrease some of the activity of the growth factor, but it will still be accessible. Or what you can do is you can bind heparin to your ECM chains, and then if you put growth factor, heparin, as we talked in the last class, has quite a bit of affinity for lots and lots of different growth factors. So, in that way the cells will essentially be able to interact with this growth factor and be able to even take it away if they need to, because this will have a higher affinity than the heparin growth factor affinity. So, so these are some of the strategies we discussed. We again discussed some examples used in the literature and saw how these systems can result in lot better improvement in cell function than let us say a covalently conjugated or just encapsulated system. Okay, so let us continue our discussion further. So, here is another example again from the literature. We will continue to give lots and lots of examples, so that you guys are getting accustomed to some of the literature as well and learn their application. So, what is being done here is they have used a polymeric system and they have used dual growth factor delivery. So, in this case what they have done is they have created a porous matrix which is written here, and in that porous matrix what they have done is they have either encapsulated their VEGF, which is the growth factor they are using, directly into the matrix, okay. And alternative to that, what they have also done is they have encapsulated this VEGF into a polymer, and then they have used a PDGF which was directly encapsulated here. So, in such a system what do you typically see, and then obviously there are lots of pores around. So, what you typically see is this. So, essentially you have a system which has pores and the polymer around it contains the growth factor that you are trying to deliver, whereas in this case you have pores, you have polymer around it, and that polymer is encapsulating another polymer that contains your growth factor. So, these are some of the two systems that they have created, and let us see what they have done with this. So, in this case now they are doing a cumulative release from this polymer of that growth factor. So, what they are seeing is if you encapsulate the growth factors into the polymer scaffold, so you get a certain type of release. So, here you are seeing that as the polymer is degrading or the molecules are diffusing, you get a certain release, whereas in the other case you can where in the case B what they have done in the case D what they have done is they have pre-encapsulated this growth factor into another polymer, right. So, this is the same case as we see here, there is another polymer which is encapsulating these growth factors, and when they have done that and then this whole polymer then gets encapsulated into another polymer which contains pores through which these can diffuse out. So, now the growth factor first has to wait for this matrix to degrade and then they can start to diffuse out. So, you see a much longer release and sort of a biphasic release in this particular example. So, this is just some of the ways these authors have tried to change the release of their molecule through these matrices. And so, here is just another example. So, what they have done is in this case they have done bolus delivery. So, essentially no matrix is used and they are trying to see whether they can generate some blood vessels, and they have used some staining to see if there are any blood vessels being generated. So, what they are looking at here is an implant and seeing how many blood vessels have grown into that implant. So, what they see, if they just have a blank implant, there is a certain amount of blood vessels density that you find. If they give a bolus injection of VEGF or PDGF, which are again two of the growth factors that we said are involved, they get certain response. So, that response goes up from 65 to let us say at max up to 100, and that this is both at 2 weeks and 4 weeks. So, it is still a marginal improvement, but does not really result in a whole lot of blood vessels being formed, which is also pictorially you can't really see many blood vessels. I mean, this is a blood vessel here, but not a whole lot of density. Whereas, if you do a sustained release, so remember how you were getting these values to be about 70, 80, where in this case now what they have done is they have sustained and a sequential delivery is being done. So, what they see now is if they only release one, it goes up to a certain value in 2 weeks, and if they do dual, it also again goes up to a certain value, and then at 4 weeks they also get good enhancement with both these deliveries. So, again the numbers are much higher. So, it is a better system rather than just giving a bolus injection. So, just one way of how you can cause the blood vessels to grow into your tissue. And then they further went ahead in a disease model. So, again in disease case the body's intrinsic capacity to heal is much lower. So, this is a diabetic model that they have used, and what they see again is if they use only a matrix, they get very little cells that are essentially vessel cells, but if they start releasing VEGF, they get certain enhancement, but still fairly marginal. With PDGF they do not really see much enhancement, but if they release both of them they see quite a long jump, almost 3 to 4 fold higher number of vessel cells being in the vicinity. So, this is again showing that if you do a sustained release, you give the enzymes and cells enough time to experience the growth factor in the matrix, move towards it, you get a much better response. So, in compared to the bolus delivery, right, what will happen if you do a bolus delivery? These growth factors are small, they will quickly clear away from the body. So, here is another paper that talked about cell demanded release of VEGF from synthetic biointeractive cell enclosed matrices. And so, again what they have done here is, this is a synthetic polymer that they have used. So, they have used a 4-arm PEG vinyl cell phone. So, what this does is, this will cross link any peptide which contains a cysteine or any thiols. So, they have created a matrix out of that, and in that matrix what they have also done is, they have incorporated peptides which are first of all protease cleavable MMPs, and they have also created peptides that are cell adhesive, and finally they have incorporated VEGF which also has a cysteine. So, that also gets bound to the network. So, this is what you see here. So, in the system you are seeing, so this is a polymeric chain. So, the PEG chain, if I use a different color, this right here is the cleavable site. So, the cells can actually break it. This is a cell adhesive peptide. So, the cells can actually bind to it. And then finally, this right here is the VEGF which the cells can come and interact with, as well as the VEGF can release over time as more and more of these bonds are cleaved and cause more cells to migrate in. So, this is basically the whole system that they have designed here, very mimicking of what you see in the tissue, where the cells can move around, the cells have growth factors to bind to, the cells have adhesive ligand through which they can actually attach to the surface, as well as they can, if they want, they can cleave the surface and further move around. And so, they went ahead with the system, and what they see here is, this is a control gel. So, there is no VEGF here, and they see a certain density of the blood vessels in this site. So, this is where the implant was put in. Then they have put in the implant and given a soluble VEGF in the implant, and what they really see is some of the blood vessels have started to grow in, but very little, because obviously this VEGF will get cleared very quickly. And then what they have done is the method that we described in the previous slide, where they have essentially put the VEGF into the implant, and what you start seeing is quite a lot of blood vessels in the implant, suggesting that this strategy is much better. And this is again pictorially represented. So, this is your control zoomed in image in this area, and so you see certain density of blood vessel. This increases with soluble VEGF. So, you start seeing these small branches coming out from some of these larger vessels, and in a bound VEGF you actually see these branches being fairly mature and in a much higher number than compared to the soluble VEGF. In fact, the whole area looks sort of green. Green is the staining for the blood cells. Okay. So, one other thing I want to quickly touch upon is some of the differences between natural hydrogels and synthetic hydrogels for tissue engineering. Again, we have talked quite a bit about both of these, and you guys have seen different examples of them. But essentially I also want to talk about further as to what can be done with these natural and synthetic hydrogels, and what to use in what case, and how do the cells interact with them. So, let us see. So, in this case I will take two examples. One is a PEG hydrogel. So, one is a PEG hydrogel, where we are looking at a synthetic material, and another is the collagen hydrogel, which in this case is a natural material. Collagen is of course one of the most abundant protein in our body. So, how do these hydrogels differ? So, let us see, let us talk about the structure in general. So, typically this goes back to what we already discussed with the PEG hydrogels. Typically, if you have a PEG hydrogel, you will get chains which are sort of cross linked either physically or chemically, depending on the functional group that is present on these PEG, mostly chemically, and there will be sort of a random orientation of them. It will be individual chains, units binding to another chain unit, and you will get a certain pore size which will be heterogeneous, but not as heterogeneous. It is somewhere hovering around their averages plus minus few microns and nanometers depending on what is the average, and you can encapsulate things in that. You can release it over time, things can migrate in if it is degradable or the pore size is big enough. Whereas the collagen hydrogels, if you use the same number of chains of collagen as you use for PEG, collagen actually is known to form fibrils. So, most ECM proteins that we find in a body are actually forming thick fibers, and then the way they form thick fibers is essentially several single chains will come together and essentially cause a thick fiber to form, and then you can have another thick fiber going through that, and then similarly you may have few thinner fibers also crisscrossing all of this, and so this is a type of hydrogel that you get with a collagen hydrogel. And so as you can see, it is a very different system between this hydrogel and this hydrogel. So that's in terms of the structure. What about the tunability? So again, the tunability is present in both of them. Synthetic matrices definitely have a lot more control, since you can define, each of them a single chain. So each of them will have a certain orientation, each of them will have control over what is the stiffness, what is the, it will be fairly uniform throughout, right. The stiffness, because these are all random chains, it will be fairly uniform, and you can change the polymer density, you can get a lot more chains in there. So, you can decrease the pore size, you can increase the mechanical properties of your system. Whereas in cases of collagen hydrogels, what you will find is the control is there, but it is not as much, right, because these fibrils first of all have really changed the stiffness in the local area. So, if I measure the stiffness of this particular gel or this particular fiber, it is going to be much stronger than these individual fibers, right. And the other thing is these are, since they are natural materials, it is sort of hard to modify them, and they sort of self assemble into these structures. So, the control is much lesser. And what about the cell maneuverability? Okay, so that is another good example here. So, now these PEG hydrogels, they do not have any cell binding site, right. So, first of all, the cells may not want to interact with it. So, what you have to do is you have to incorporate cell binding sites. And what I mean by that is that if the cell comes in and wants to interact with this, at this point the PEG chains do not have any ligands. So, unless I put a ligand, let us say RGD that we talked about in the previous case, the cell is not able to bind to this. Only if there is RGD or some other sequence, the cells can actually bind to these matrices. So, that is one. The other is, what if the cell wants to move around? Now, the way the system is set up is these pore sizes are fairly uniform, because these are single chains, and what you will find is typically to form a hydrogel you need quite a bit of chain. So, these pore sizes typically you do not get, these pore sizes typically are always less than 100 nanometer. Now, when we talk about cells, we are talking about at least mammalian cells, we are talking about mammalian cells which are typically always greater than 5 microns. So, now if I am saying the pore size is 100 nanometer, which is fairly uniform plus minus let us say 10 nanometer or 20 nanometer, these cells can't really move through that. They are sort of locked into all kinds of polymer chains crisscrossing them, and they can't move anywhere if you have encapsulated these cells inside. So, if you want cell maneuverability, what you have to do is you have to also incorporate the protease cleavage size. Which basically means that now if I change this this sequence to let us say these green regions, which are degradable by certain enzymes that cells have on their membranes or can secrete, only then the cells can actually start degrading this. So, they will break it, essentially breaking the whole chain, and then they will be able to start to move out. And so, that is one of the issues with these synthetic hydrogels, because you have to take care of all of these things before it becomes cell compatible, because essentially if you want the cells to be fairly happy in that system, you want some maneuverability of the cells in that system. So, you will have to do all of that, and even then it may not be enough, because let us say a cell wants to move at a much faster rate, but it first has to wait for the cell to degrade the protease cleavage site, which could be at a much slower rate, or vice versa. Maybe it degrades too much and now suddenly the cell has nothing to bind to, it is completely in now a pore, it is looking for active site and everything is getting cleaved. So, all of these issues start to come in when you are talking about synthetic materials, and you have to do a lot of research to sort of figure out what is the rate at which you want the these peptide sequences to cleave, and that will vary with cell to cell type. And not only that, it will vary from application to application, and then not have a very control on that system in terms of the cell happiness or the cell compatibility with that system. Now, let us compare to what happens with the collagen hydrogels. So, we already talked about the structure and the tunability. Structure is described here. Tunability, you get a better response from synthetic hydrogels, but when it comes to the cell maneuverability, all of these itself have cell binding sites. Most ECM proteins will have a certain cell binding site. So, this could be collagen as is written here, or this could be fibronectin, could be laminin, could be something else. All of these have cell binding sites. So, these will bind to the cell, so that is not required. What about the movement of the cell? So, now what you have done here is, because these fibers have sort of self assembled into big fibrils, the pore sizes are very different. So, what you will find is typically the pore size in such a gel can be easily changed from let us say few nanometers, let us say 1 nanometer to all the way to tens of microns. So, certain regions will have large pores, certain regions will be fairly compact as you are seeing here. So, this you are talking about less than 1 nanometer gaps, and this you are talking about huge gaps of let us say 10 of micron, and because of that you do not really have to worry about cell maneuverability. So, when the cell wants to move around in the gel, it does not have to wait for some sort of degradation kinetics to happen and move around. So, this can happen by whenever the cell wants to move around. And not only that, these are natural materials as we just talked about. So, the cells have capability to actually go ahead and degrade the collagen fibrils if they really want to, or deposit more. So, the cells can actually come and deposit more collagen, and that collagen will then interact with this collagen that is already present, and so the cells can move around easily. cells can remodel the network easily, and then cells can also bind the network easily. So, you can see all of these are sort of advantages of a natural system that you get, because you do not have to worry about now making another component that has cell binding site, you do not have to worry about making another component that has cell cleavage site, that is all inherently present. At the same time, there is obviously drawback that the tunability is fairly low. So, this is cell maneuverability is very high, in this case it is low, tunability is low in this case, high in synthetic gel, and the structure is obviously different, and the control on the structure is sort of high here. Structure, if I say control, is high here and lower here. So, these are some of the advantages and disadvantages of both synthetic and natural hydrogels systems when you are looking at the cell level and how does the cell interact with it. So, just something to keep in mind, and something to consider when you are choosing a material for any application that you want to use for in vivo or in research. So, it is important to consider these factors in mind. Okay. So, we will stop here and we will continue further in the next class. See you then. Thank you.